Hi, I'm Jack. I'm an ecologist. I actually started out my career as a butterfly specialist. I worked on butterfly distribution patterns in Africa for the Natural History Museum in London for a long time, until somebody thought it would be fun to send me to Antarctica to find bacteria that produce proteins that make ice cream taste smoother, which was the most noble thing I ever did, and it's been downhill since then. My primary goal has been to explore ways of engaging ecology in medicine, right? For the last 10 years, so for the last 9 years, I was a professor of surgery at University of Chicago and ran the Microbiome Institute there. And now I've been lured away to the wonderful beaches of San Diego to explore that same phenomenon in a new place. And I work very closely with Rob, and so we have a lot of the same foibles of a lack of attention and focus to anything in particular, and a desire to try our hands at everything. And that's a really, it's a fun place to be. I don't know if you see from the slide, but I'm actually currently in the Department of Pediatrics and Oceanography at Scripps Institute of Oceanography, which makes me the world's first pediatric oceanographer, or oceanographic pediatrician. I don't know, I could try both. I do work with baby dolphins though, so that is a thing, and it is fun. And we look at how microbes can be used to predict disease states in some of the animals used to, dolphins and sea lions and marine mammals that we try and protect along our coastlines by taking samples at the beginning and using a lot of data regression and then some fancy AI. We can actually start to derive predictions about the risk of the development of, say, a viral outbreak in that population. And that is, for most ecologists, a wet dream, right? That's what we've been trying to do for years, be able to actually apply what a clinical group would have been able to do for years. We can apply that into an ecological setting and try and predict disease outbreaks and understand the movement of animals. That's phenomenal. And I bring that up as a key point, right? We are trying to marry those two relationships. We've built a lot of our human microbiome research on prior research that came from the field, right? So the trees and our soil work and our ocean work and work in rivers and lakes, and that really did help to spur microbiome research in human health along dramatically. Well, now it's coming full circle. The developments in the clinical sector are now being reapplied back into the environment. And that is a continuum that we want to continue to grow, right? We want to harness that in order to make both worlds more significantly better. I have a number of disclosures. And I'll make this point that, for example, with one of them, BiomeSense, I'm extremely keen to see the rapid translation of some of the findings that we've developed into those clinical settings, both technological advances as well as new diagnostics and therapeutics. And I think it's immensely important that we embrace that reality. And that's something a lot of faculty aren't necessarily geared up towards doing. But it can make a substantial impact on obviously our patients, but also on trying to develop strategies that will be more effective in building programs of research that will have that outcome. If you have the goal in mind, you can figure out a way to get there, and that's the most important component. So our microbiome is obviously incredibly important to us, and one of the primary components that I like to think of in this respect is those microbes that are in our body that are playing a significant role in a key aspect of our health, right? I was just at the SoCal Microbiome Conference where I saw a wonderful talk by Lars Bode and several others that demonstrated that, a phenomenon which I was very used to. I've got two children. When both of my children were young, they were both breastfed, both vaginally delivered. During breastfeeding, I would have sometimes 2 or 3 nappies to change a day. And I say I because, yes, I was mostly around when they were very young. I tried to be at least. And most of those nappies were actually, diapers, to use the American vernacular. Most of those diapers were actually pretty wet and mucousy, right? And I had just completely glossed over this. It turns out that a wet mucousy diaper is actually a sign that most of the human milk oligosaccharides associated with breast milk weren't properly digested, weren't properly fermented in the gut. So you end up with a lot of them being directly excreted. And when pushed upon this, why would this phenomenon be so? David Mills from University of California, Davis came up with a very specific proposition. He said, well, they don't have any Bifidobacterium infantis in their guts. And Bifidobacterium infantis is highly prevalent in the guts of many children growing up in developing nations, or nations which haven't got as much acculturation, as much desire to live in this kind of nice environment. That bug is missing in many American and Western children's intestines, and increasingly so in children growing up in China and Southeast Asia. Now, why is it missing? We don't know. I have a lot of theories, and I'll highlight a couple of those today. But the very premise that we could take for granted that our kids were healthy and everything was going fine, when there was clear evidence in the poop that things may not have been going fine, is testament to how rapidly our ideas are evolving and being put into improving our understanding of what we can see with our own eyes, but also the power of the things that we leave behind every morning when we flush the toilet and throw away as a really important aspect of how we understand our health. And so these microbes we know are important. And we have 1,000 other stories, and you heard some of them yesterday, and I'll tell you a few more, that are incredibly compelling as ways of developing these organisms as therapeutic interventions, let alone diagnostics, but therapeutic interventions. That's where we really want to see it. And Ian was mentioning fecal microbiota transplants, right? We've also seen very recently one of our colleagues at UCSD, Stephanie Strathy, building upon her pioneering drive to keep her husband alive from a pathogenic infection, leading to the use or the reintroduction of phage therapy back into the US healthcare system. Something that was very prevalent 100 years ago and got wiped out due to the fact that phage therapy was thought to be a communist threat on our population, and we should just all embrace antibiotics. That kind of reimagining of the clinical strategy, I think, is amazingly important. And if you haven't read her book, The Perfect Predator, you should. It's also about to become a film in maybe a year or so. That's very cool. So a lot of my work stems on trying to understand how the microbiome interacts with the immune system and what that means when it goes wrong. I like to think of the immune system as a national park warden or a gardener, right? It's not there to act as an army to go in and wipe out any dangerous pathogens that occur. That is a role, but that's the role of any national park warden as well. Its main aim is to act as an ecologist to shape and manage that ecosystem. The ecosystem inside your body, the cells that make up the garden. And like any good gardener, she has to manage the crops that she wants to grow, but also to make sure that the weeds don't overrun those crops and damage the overall balance of that system. Maintaining balance is really the auspice of ecology, right? Ecology is the study of balance. And in this context, understanding that intricate relationship between the microbiota and the immune system has become paramount to how we understand health. And remember, these are two sides of the same coin. The immune system and the microbiota are both intricately dependent upon each other. If the microbiota changes, so does the immune system. If the immune system changes, it changes what organisms can live inside you, and therefore it changes the microbiota. And those two pieces are not necessarily cause or effect, they're both. They are in a delicate feedback loop horror story of complexity. Trying to understand that relationship is challenging, but it's also incredibly rewarding when you actually find individual components to understand the mechanism, and I'll tell a story about that at the end. So microbes are little chemical factories. I use this quote from Jorge Luis Borges. I'm not entirely sure he ever actually said this, but as an author, his literature was defined by the intricacies of human associations. Everybody had an influence on everybody else. So if you understood, you saw a character in one of his novels, you'd find out that that character affected something in another novel much later on. And that relationship is key to our understanding of the microbiota. We test this association with what we call microbiome-wide association studies, akin to a genome-wide association study, or a GWAS. It's just trying to figure out which microbe, which functional gene, which metabolite they produce may be associated with a disease. We call this a statistical association study. And it's been at the cornerstone of helping us to identify that microbes are important in the first place and can describe some of the variants in many of our disease states. But it's also absolutely essential for identifying those diagnostic and therapeutic biomarkers. Without that study on a large population of people and using it to divide you all up into groups, we wouldn't be anywhere near as close as we are today to seeing some of the first therapeutic advances in microbiota become clinically relevant as an FDA-approved strategy. Again, the only one we have now is fecal microbiota transplant, but within the next 3 or 4 years, I think we're going to see a huge number of these trials move into phase 3, pass phase 3, and into approval very quickly. We also know the microbiota can explain a huge amount of variance in many of these different disease states. So this work from the Weizmann Institute identifies that microbiota alone can play a significant role in describing the variance in glycemic status in a population, or the waist circumference, or lactose consumption potential. The ability to combine basic clinical measurements alongside the microbial variants, the population of bacteria, their functional traits, their metabolism, and adding in human genetics, and combining all of those together is an epidemiologist's wet dream. The opportunity to grab all of that data and build models which are actually effective at describing variants in the population, that's huge. And that is at the baseline of everything that we're trying to achieve. Trying to achieve a fundamental understanding of why our patients respond differently to different drugs. Why certain people with a particular gene for Alzheimer's never seem to come down with the disease, while others seem to have very rapid onset. What describes that variance? And we think the microbiome is a significant effector in that relationship. I want to touch upon what I would as often referred to as dysbiosis, and use it as a segue into trying to describe to you how we use these phenomena to try and understand disease burden. So I have 2 different scenarios here. This is from a wonderful review by Bindlos et al. In 2018 in Mucosal Immunology. But the basic premise is everything is in equilibrium. Everything is touching everything else all the time. It's ecology, right? Under a homeostatic environment, we have balance. We have equilibrium. We have microbes living in a oxygen-poor environment, like the gut, which has very low oxygen potential, referenced there in blue. And we have bacteria there that don't like oxygen, obligate anaerobic organisms. And many of those obligate anaerobic organisms are extremely good at fermenting the fiber in your food and producing things like short-chain fatty acids such as butyrate. Butyrate is incredibly important as a regulator of the beta oxidation activity in the colonic cells, leading to maturation of those cells, whereby they actually start to consume more oxygen from the blood and further reduce the oxygen potential in the gut, because now oxygen's not bleeding from the bloodstream into the gut, which is actually a problem. In that context then, microbes are there because of the way the gut is, and because of the maturation of those cells, but they're also causing the maturity of those cells. And that's a positive feedback loop. If everything's going well, everything stays in balance because of that cyclicity. And understand that cyclicity means removing yourself from the dogma that X causes Y. Because now X causes Y, causes Z, causes X, causes Y, causes Z, causes X, in some kind of virtuous cycle, which we have to embrace if we want to understand health. Interruptions to that cycle can lead to what I would refer to as dysbiosis. The very appreciation that when something goes wrong, or when something in that loop is disrupted, it affects everything. So in this case, we're giving a patient an antibiotic, a non-absorbable antibiotic, vancomycin maybe, and it's eradicated a lot of the microbial biomass. And there was a latent organism in there, a gamma proteobacteria, which can be facultatively anaerobic, that is perfectly happy with a higher oxygen potential in the lumen of the gut. Now what we've got is an overgrowth of these types of organisms. None of them are digesting fibers in the way that our obligate anaerobes were before. these facultative anaerobic gamma proteobacteria are in fact often producing other compounds like hydrogen sulfide. And when they do that, that hydrogen sulfide can cause irritation and inflammation in the gut. But it's also because of a lack of those butyrates and short-chain fatty acids like butyrate that are being produced by the obligate anaerobes, our colonic enterocytes are no longer maturing. And when they're no longer maturing properly, we get leak of oxygen into the gut, which exacerbates that cycle. Now we've entered a new stable state, not a healthy stable state, a dysbiotic, a disequilibrium stable state, whereby the gut is no longer potentially positively interacting with the microbiota. In fact, now we've got a negative association loop. And understanding that's a secondary loop is important. It also means if every aspect of that loop is a cog, if we swap out one of those cogs, we may be able to drive the whole system back to equilibrium. And the only major thing we can do in that system is to change the microbes that are present. If we can change the microbes that are present, we can re-engage the positive feedback loop and have a big impact upon health. And I know John's here, and John and Mary have both talked about mental health, so I don't have to go over this too much. But we think that there's a very significant association between disruption of microbiota and clinical health. As referenced by some of the fecal microbiota studies, of which Professor Cryan has been a major part of, understanding that we can take a microbiome from a depressed person and put it into an animal model and see changes in behavioral state has been absolutely essential as a pre-indicator of the role of those microbiota in that relationship. What's not obviously clear, and it's becoming more clear with more studies, but what's not obviously clear is the mechanism behind that relationship. What's actually causing the turnover of microbial activation of these dysbiotic brain states, where our mental health is impacted by a disrupted microbiota. Work that we did with folks such as Phil Stramwitz and Kim Lewis up at Northeastern University identified associations between, for example, the disruption of the connectivity of the prefrontal cortex leading to activation of a depression-like symptoms. This kind of disconnectivity is highly associated with major depressive disorder and is variable and undergoes changes over time. We're able to identify Bacteroides, a genus of the Bacteroidetes, certain species of Bacteroides which are incredibly good at producing gamma-aminobutyric acid, and they did that under a low pH in the gastrointestinal tract. And when they did that, they are positively associated in terms of their relative proportion across a population with an improvement in the connectivity of the prefrontal cortex, which is associated with a more positive mental health state. And that relationship led to using those particular organisms in clinical and preclinical trials to determine if they could have an impact upon animal health. And those trials are ongoing in our lab at the moment, but we can definitively stipulate that when introduced into a rat model, the rat model shows reduced markers of anxiety. Now, okay, no rat is, as far as we're observed, anxious, and you know, we have to hold up that, I think John would agree with this, right? Most of our animal models for mental health are terrible, but, and Mary would probably agree that most of our models for mental health during pregnancy are even worse, but we are getting there in terms of being able to identify organisms that can have an impact upon this virtuous positive feedback mechanism. We also understand that microbes can play a role in other mental health disorders or degenerative disorders such as Alzheimer's. And in a study that we just published with a group from University of Chicago, Sam Sisodia, we're able to determine that when animals are treated with an antibiotic cocktail, we have an impact upon the microbiota, but we can also have an impact upon phenotypic traits associated and clinical traits associated with Alzheimer's. In this case, the deposition of beta amyloid in the brain tissue. In fact, when we have here a male vehicle with no antibiotic treatment, and you see all those little yellow dots in the brain tissue. I don't know if you can see them on the screen, hopefully you can. That's beta amyloid being deposited. If we give the male mice antibiotics, we see a significant decline in the number of beta amyloid plaques being deposited in the brain. suggesting that there's a relationship between the presence of the microbiota and the actual disorder. We've repeated this in germ-free animals, and we see the same phenomenon. I.e., if you reintroduce the bacteria into the germ-free animal, it has an effect. Antibiotic treatment on a germ-free animal has no effect upon this phenomenon. Interestingly, female animals are less responsive to this kind of therapy. We see a no significant difference in beta-amyloid deposition pre or post antibiotics, unless we wait a little bit longer. And this is data I don't have on me, but if we leave the animals under their beta-amyloid deposition state for a longer period of time, the female animals will get a significant increase in beta-amyloid deposition, which can be partially ameliorated with the use of antibiotics. Again, this is a very big method, right? I can't think of the word anymore, but you know, a very harsh method. We are not using a targeted strategy to remove or add specific organisms. We are just going blanket kill strategy, right? We're going to wipe out as many of those organisms as humanly possible in order to try and effect a difference. And that particular strategy is proving to be quite powerful in helping us to understand that differentiation. And none of my videos work because they were all not transferred over. But I do want to move on to trying to explain what I think is potentially going on in terms of our lifestyle that could be having an impact on some of these effectors. Every single one of you in the room right now is releasing around 38 million bacterial particles into this conference center, right? Every hour. In fact, your hotel rooms right now look very similar to your bedrooms back at home, microbiologically speaking. If I swabbed the floor there today, I wouldn't notice any significant difference compared to your bedroom, other than the absence of whoever you share that bedroom at home with, and potentially the presence of whoever you're sharing the bedroom here with, but we won't go into that. But that relationship is potentially extremely important, right? Is there any other biological agent in this room producing microbes into the environment? No. There's no animals, there's no plants, the air is being filtered, all the doors and windows are closed. The only source of microbes into this space is your microbes. Now let's say we took an infant who was just born and you got a healthy dose of her mother's microbes, sure, and then we just placed her in this room and expected her to grow up. What kind of immune stimulation would we expect that she would get? Not much. She'd just get human skin, human oral, and potentially human urogenital associated bacteria as her primary source of exposure. Now, is that the way humans developed? Is that the way our immune systems evolved? Is that what we expect to see in terms of immune stimulation? No. In fact, well, not based upon the evidence over the last 2,000 or 3,000 years. Our bodies were being constantly bombarded by microbes. Yes, we've got good at controlling some of that bombardment, such as vaccines, which are immensely important. But we've actually, in our drive to remove pathogenic disease from our society, have nearly totally eradicated our exposure and potentially leaving us open to many immune dysregulatory effects of not having that appropriate exposure in the first place. And there are many ways in which microbes can actually interact with you, and you can interact with microbes in these built environment spaces. Airborne is one of the key ones. The bacteria inside my oral cavity and inside my lung are being constantly expelled as I talk, and you're probably getting way too many of them, I apologize. But also, so I'm leaving this constant stream, but also I'm breathing in tens of thousands of microbes with every breath. A litre of air can contain somewhere between 500 and 7,000 or 8,000 cells, depending upon the environment. And so that inhalation of breath is exposing my mucosal immune system to microbial adjuvants. We also know that the things we touch, the surfaces we touch with our hands, the seats we touch with our bodies, I was going to say our buttocks, but that sounds weird, are also a source of transmission. If you think that the clothes you're wearing are stopping microbes from moving between you and the chair, you obviously have a very poor appreciation of the size of microbes. They're very, very small. And most of your clothes are like loose fishnet stockings to them, right? They just pass straight through. So the reason pulling down your sleeve so you can open the door handle in the bathroom is kind of irrelevantly pointless. It makes no difference to transmission whatsoever. If it makes you feel better, then the psychological phenomenon is working. But that relationship with your environment is continuous, and that exposure is incredibly important, we think, to developing immune system responses. But there are other aspects. So, for example, in the building that we're in now, it's very dry, partly due to climate, but also due to controlled air conditioning, which removes moisture from the air. But for the vast majority of our history, and if you've ever seen any of the fallout from Hurricane Katrina, you'll notice that when things get wet, stuff grows. And when stuff grows, it produces metabolites. And some of those metabolites are volatile and get into the air, and those can have massive impacts upon our health. One of the key indicators for respiratory diseases in the inner city is hidden fungal growth in the walls. A lot of it you can't ever see, it's just there, it's hiding behind the wall, everything looks perfectly, or the landlord put a fresh lick of paint on the wall and you never see it until it starts to bleed through and the mold starts to produce things which will inappropriately affect your immune system. There's also a wonderful story about ghosts, and Ian knows I like tangents, but I'll just tell us one briefly. There are fungi in old homes that can produce a psychotropic effect upon the neurological system of a human. Right, so the idea, and it's probably completely wrong, is that some of these, if they build up in an old creepy home and you open the door, you get a sudden burst of these metabolites in the air which cause you to have audible and neurological stimuli and hallucinations. Right? So maybe microbes gave us all the ghost stories imaginable in our entire history. Wouldn't that be kick-ass? Another good reason to thank microbes. The entire premise of Halloween is probably based on some kind of microbial hallucinogenic activity. To understand this phenomenon of microbial transfer and potential microbial threats to the immune system, we went where we'd expect people to be under immune system distress, a hospital. It's the Center for Care and Discovery in Chicago. And we did 365 consecutive days of monitoring of this environment, 2 days before it became an operational hospital environment, 2 months, analyzing just the room samples, the surfaces in the nursing station, the air and the water. And then for 10 months post-opening, adding in the microbial sampling of the patients themselves and of the hospital staff. And that provided us with a map about how things are actively moving between us and the environment. But it also provided us with an opportunity to understand the evolution of an environment which previously hadn't had any human occupants of merit. Vast majority of the microbial organisms that came from people in that building prior are eradicated due to several rounds of extreme biosterilization of the environment before it becomes an operational hospital. So before they move anybody in, they basically bleach the entire inside of the building. Now, I want to point out something about bleach. If I sterilize that top right now with bleach, as soon as the bleach had dried out, all the microbes from you people and me would be landing on that surface, and some of them would survive, right? So any kind of biosterilization event, just go ask NASA about what happened with some of their probes they sent into space, doesn't work. Things will always find a way to land on that surface and grow. Anyway, in this context, we were able to map that relationship and build up a picture of what happens. On the left, we have a principal coordinate plot. Every dot is a microbial ecosystem, and if the dots are closer together, they're more similar. And the big differential there is the samples in blue are before the hospital became operational, and then the samples in red are after the hospital became operational. And you can see that there's a big shift towards an entire new microbial community as the hospital becomes operational. The main driver behind that is an increase in bacteria associated with the human respiratory tract and skin. Again, those 38 million bacterial cells you release every hour into your immediate vicinity are colonizing the hospital as you colonize it. That's an important precept to understand. We also were able to identify more disturbingly maybe, that over 95% of the organisms you left behind in the hospital or you leave behind here now die as soon as they leave your body, or within a few hours. They are not adapted to living on the cold, dry, sterile environment of a building surface. So they often just eradicate. But the ones that do survive have selective pressures that drive them towards either the acquisition or increased in prevalence in a population of genetic traits which allow them to survive. And in some cases, those survival traits come along with other traits that allow them to survive other threats, like antibiotic resistance. What we actually saw is that the main indicator in a year of observation of a hospital for the buildup of antibiotic-resistant bacteria in that space was not what antibiotics were being used or administered to the patients. It was purely those organisms that could survive in the cold, harsh reality of a hospital environment. They were building up antibiotic resistance at very high levels to antibiotics that weren't being used in the hospital or in the hospital rooms to treat those patients. There was no correlation there at all. And this suggests that stress in the environment is a key precept which allows us to understand why things are developing. NASA's very interested in this because if you stick an astronaut in space and she has to travel for 3 months to Mars and 3 months back for a 3-month mission, she's trapped in a capsule whereby her microbiota is going to be being weaponized by the capsule environment, because the only things that she releases that are going to survive are going to be things that can survive, and those ones might be able to attack her more readily if she becomes sick. And that paradigm is potentially incredibly important, and we just started a 3-year grant to explore that more readily. I'll skip on a little bit. We wanted to be able to understand what happens to those organisms when they land, when they leave your body. And the vast majority of them will only grow if there's moisture around. And I mentioned this before with Hurricane Katrina, etc. In the left here, we have bacterial communities, and we can see that if they're dry, the diversity doesn't change. It fluctuates, but it doesn't change too much. If the environment's wet, they're in blue, we get a significant reduction in diversity as we get overgrowth of particular organisms which can survive and can thrive in the moisture. Same is true for fungi, but fungi tend to grow irrespective of whether we add water or not. Fungi are incredibly good at pulling moisture out of the air. They're incredibly good at this, which is the reason plants like being associated with some fungi. It helps them to survive drought stress. And so even on a dry surface or a relatively dry surface, they're pulling moisture out of the humidity in the air and using it to grow. Interestingly, the types of chemicals that these organisms were producing varied, but not in terms of the absolute abundance of chemistry. Chemistry stayed relatively stable over time, irrespective of moisture development. We also demonstrated, quite interestingly, that bacterial and fungal diversity is well correlated, and sometimes, so if you have more bacteria, you often have more fungi. Interestingly, Bacillus and Pseudomonas were anti-correlated, suggesting some kind of competition. When one grew, the other didn't. And things like Eurotium, a fungus, grows very well in the absence of water as it pulls the moisture out of the atmosphere, but Penicillium cannot do that. It only grows when you add moisture. Ecologically, that's fascinating. Biomedically, it's potentially important, as I'll explain in a minute. We also find that the fungi are producing lots of compounds which tend to clear the path around them of bacteria. They actually view, in this nutrient-poor environment, bacteria as being a threat to their survival, and so they tend to produce compounds which can kill them off. And we find that Bacillus and Pseudomonas are actively fighting. So Bacillus is producing azoxystrobin, which is an antibacterial targeted towards Pseudomonas. And Pseudomonas is producing scopolectin, which has the opposite effect. They are actively fighting for resources, so they enter into warfare. What's very interesting about this phenomenon is we are currently engaged in a clinical study in the Rady Children's Hospital down in San Diego, where we're adding Bacillus back into the environment, actually actively adding bacteria back into the hospital environment as a way to control Pseudomonas aeruginosa infections. Because when we add the Bacillus in, the Pseudomonas levels plummet. And if you have an active Pseudomonas infection in your hospital, you don't want that to grow on surfaces. And so far, we can't figure out a way of stabilizing any of the antibacterials which kill off Pseudomonas other than covering the entire surface in copper, which is incredibly difficult, very expensive, and has, currently, as we're seeing, actually quite a high rate of resistance. Microbes are actually finding ways to resist copper as an antibacterial. Pardon me. Interestingly, when relative humidity goes up, and this was in Chicago, the microbial sharing goes up. So people share more microbes when they, even if they're not physically interacting, if there's more moisture in the air. Things can survive longer outside. This is probably a good reason why we see some increase in bacterial transmission during these events as well. And staph were a greater source of bacteria to patients than patients who had staph. So on the right of this Bayesian map, where every node is a codependent scenario with other nodes, allowing the tracking of the movement of microbial elements between those nodes. We can see that the staff on the left were nearly a contiguous source of bacteria to the patient, but not the other way around. This, we think, is an artifact of observing staff. We were observing them very closely, and so they washed their hands very rigorously after visiting every single patient. So that while they were still a source of microbes to the patient, the microbes from the patient that they picked up were removed when they washed their hands. Good indicator that washing hands works and reduces transmission of microbes between patients, but also difficult if we want to be able to track the actual transmission event. We wish people would have been less diligent, although it might have killed somebody, so that would suck. We also know that people that physically interact share more microbes. So when patients and staff physically interact, we know they do. But even if you take, for example, this is three people moved into a flat in Chicago, and two of them became intimately involved, and the other one got left out. Wasn't that kind of flat. And in yellow here, on the left, for example, we're looking at all the microbiome samples. You can see yellow is the lodger that is living with this couple, right? And the couple are in red and blue, and they are physically more interactive, and therefore they share more microbial similarity. When you physically interact with someone, you share more bacteria, and therefore you increase the degree of overlap. Other than in the nose, on the far right there, you can see the nose. For some reason, the couple haven't quite figured out how to interact with their nostrils. But you know, that paradigm still holds true. And we can track the microbial distribution patterns quite effectively, right? We can see microbes moving between different surfaces. So for example, here we have a young family, and we can see microbes moving between their noses and their hands and their feet, and also microbes moving from, say, the floors to their feet or surfaces to their hands. And you can't really see it very clearly there, but hand number 1 is a young child. He was only 6 months old, and his microbiota on his hands are more physically interacting with the floor than other occupants, because he's crawling on the bedroom floor and physically interacting with it more frequently. Interesting, when we add dogs into that scenario, everything changes, and we get a significant increase in microbial sharing, not just because of the dogs, but between occupants as well. And work that Rob Knight's done demonstrated that couples that have a dog in their home are more microbiologically similar overall. This is a study of 178 couples than couples without a dog, suggesting just the presence of a dog increases the degree or the potential for you to share microbes, and therefore transmission events, potentially, in this case, beneficial ones. Microbial transmission can, in some cases, be immunologically beneficial. And we actually have a very large study in baboons where we're demonstrating this effectively now with Beth Archie from Duke University. But interestingly, in this case, couples with a baby share less microbes than couples with a dog. So if you think that microbial similarity with your respective other might be important, then you should rescue a dog, not get a baby. It's not going to save your marriage. And my wife took this to heart and made me rescue Captain Bo Diggly over here. I did not name him. But interestingly, this again ties back into hospitals and space. I'm making an audacious statement that if we can't put more dogs in the space capsule as potential ways of improving immunological health of the astronauts, maybe we could just capture the dog microbe essence and use it in the environment in order to improve immune stimulation. And I know that sounds like bullshit, but in my mind, it might actually be an effective strategy at helping to mitigate disease burden in these populations. Homes with dogs in them have less antibiotic resistance in the building, and also children that physically interact with dogs under the age of 1 have a 13% reduction in the likelihood of developing allergic disease and asthma. So just having a dog in your home is beneficial, and you'll notice I haven't said anything about cats. We can also do microbial forensics. This is a bit of fun, not necessarily health related, but every single, we have different surfaces here. So kitchen counter up there on the top right, for example, and bathroom doorknob, and then floors, and then front doorknobs and light switches. The x-axis is time, and the y-axis is the percentage of bacteria that were found on that surface that originated from one of 6 occupants. In red, we have person 1, who's a 25-year-old man living with his mum and dad. His dad is person 2 in orange, and his mother is person 3 in yellow. And you can see the severe gender imbalance in this home if you look at the kitchen counter, and the fact that often 100% of the bacteria on that counter surface originated from the mother. until she goes away for a very short period of time, just 2 days, and then the father's microbiome explodes there at the end, that orange peak, as he desperately fends for himself trying to make toast, probably. On the bedroom floor, we have a weird peak from the young man in red there. We're not sure what's going on. Other than that, the vast majority, I won't go into it. Other than that, the vast majority of microbial interactions, microbial organisms are sourced from the dogs. And you have to have dog bacteria on the light switches and doorknobs because you act as a transfer agent. You pet the dog, and you have dog bacteria on your hand, and you touch those surfaces so you can transfer things. And this means that we're trying to figure out ways to better manage that environment. And so, you know, adding dogs, maybe cats, I don't care about cats, but might be important. Reducing surface moisture could be important. We are developing airborne sensors. We have a patent associated with that, and we're now affecting it in fecal matter, but you can also do this in airborne environments to detect microbial organisms and their products automatically. Adding plants, increasing ventilation, potentially increasing access to cows. And I can talk ad lib about that for ages, but I just wanted to choose one more fun nugget. We worked extensively on trying to understand this phenomenon in population scale studies, and we worked very closely with the Amish and Hutterite populations in a study with Carol Ober at University of Chicago and Donato Bocelli at Arizona. In this study, we were able to access these populations which have the same hereditary link. They both originate from communities that came over in the 1800s. They both have the same genetic predisposition towards the development of asthma. They are both predisposed to asthma. And they live very similar lifestyles in terms of lack of technology. At home, there's no televisions, no phones, no iPads, etc. The only difference is how they approach farming. So the Amish live on the family farm. The kids are the front door's 50 feet from the barn door. The kids work on the farm before and after school. It's a highly stimulating environment for a child. The Hutterites don't. The children are separated, the housing area is separated from the farming area, and only the adult males are allowed on the farming area, so the kids get no exposure whatsoever, male or female. And we can see that in their immune systems. In yellow here we have the Amish, and in green we have the Hutterites. And the Amish have a lot more neutrophils, so those phagocytes running around, munching up and eventually dying as they identify foreign objects. You're exposed to more foreign objects, as you would do if you were milking cows and running around inside a barn. You're going to have more of those neutrophils in your system. The Hutterites have more eosinophils, pro-inflammatory eosinophils specifically, as you know, you'd expect for a population that has a very high level of asthma. And I didn't mention this, the Amish have 4% asthma in their population, half the US average. The Hutterites have 25% asthma in their population, massively elevated levels. Interestingly, the neutrophils in the Hutterite children look older. They're expressing these CXCR4 and CD11B and C cell surface markers, and we hypothesize that this makes the neutrophils more sticky. They bind up together in rafts, and when they finally see a foreign object, they produce a cytokine cascade, causing a pro-inflammatory response. That's the hypothesis. To test that hypothesis, we took dust from the bedrooms of Amish kids and Hutterite kids, sneaking around like the Grinch at night. No, we didn't. We just did it in the daytime. It'd be weird if we did. But and we took that dust and we made intranasal installations of it, and basically a little liquid that we ejected into the nose, into the nasal passage of mice that had been sensitized to evalbumin. And so you've got a mouse sensitized to evalbumin. We either gave it dust from an Amish bedroom or dust from a Hutterite kid's bedroom, and we see the effect. And indeed, my marker probably doesn't work there, but there it is. You can see here in terms of neutrophils, yeah, there it is, that the children that got Amish dust here in orange have a massively higher level of neutrophils, as the kids did, right? And a significant reduction in eosinophils, again, just as the children did, suggesting that the Amish dust is protective against eosinophilia as a byproduct of evalbumin exposure. And indeed, the Hutterite dust had no such protective effect. So this could lead you to the immediate assumption, well, maybe I should just go down to every Amish farm and bottle it up. The question you have to ask yourself is, how much do you have to be exposed to, at what age, and for how long? We have no idea about that data, and that's the reason we can't just take a child's face and rub it in the side of a cow and hope for the best, right? We have to figure out more effective strategies at delivering it. And so to do that, we started taking poop from the Amish kids and the Hutterite kids, just to see if if that immune exposure, maybe it was changing something about the chemical metabolism in the gut. And so indeed, when we take Amish poop and we put it into the gut of an animal, it shows a significant reduction in pro-inflammatory eosinophils, a significant increase in neutrophils, and an improvement in reducing those pro-inflammatory eosinophils, and an improvement in the abundance of what we call homeostatic eosinophils that are anti-inflammatory. And we can see, when we look at the serum of those animals, we see a significant increase in just two particular traits here. Up at the top in blue, those are the metabolites that are enriched in mice that have been populated with Amish poop. That's 3-phenylpropanoic acid and indole-3 acetic acid. And we actually hypothesize, we've done some germ-free studies now to back this up, the things like indole-3 acetic acid produced by bacteria in the gut are actually stimulating eosinophils in the lung via the AHR1 receptor to become more homeostatic, in the fact leading to an increase in IL-22 production, which drives down inflammation, and a reduction in dendritic cell activation and type 2 cytokines. So we actually hypothesize that the immune stimulation that you get from breathing in all that cow dust is changing the bacteria that live in your gut. Remember, they're two sides of the same coin. And that those bacteria are now producing different metabolites, like indole-3 acetic acid, which are actively and disparately from the gut, influencing immune cell activation and changing the ability of those immune cells, or the potential of those immune cells, to respond negatively or positively to antigenic exposure. Again, wonderful cyclical feedback mechanism where everything's interacting. You need ecology to study behavior and health. And we can also find that the things you breathe in are incredibly important in helping us to understand those phenotypes. So when we look at the microbes on the mucosal surfaces in the lung, we're actually able to define fungal and bacterial biomarkers, or diagnostic biomarkers of different asthma phenotypes. So we can actually delineate them into those that will be more pro-inflammatory response, those will be lower pro-inflammatory response. And again, this is not in farm-dwelling people now, this is just people in cities. So by dividing up people and separating them into individual groups, and then using that information to drive our ability to intervene in those different populations, we might be able to have an effect on eradicating asthma within the next 5 years. And I know Rob may have talked about the American Gut Project, did he, Neil? Ian, did he? Yeah, okay. This is my microbiome. It was in Chicago, now it's down in San Diego. It's a snapshot, it's a bit of fun. But if we combine all those snapshots together and put it into a publicly accessible database, it can have a big impact. And we know that most people, most companies out there looking for microbial therapeutics and diagnostics are leveraging the American Gut Database as a freely available resource, admittedly crowdsourced from the people that provided those samples, as a way of determining whether their diagnostic biomarker is distributed differently in different populations. And the bigger this database gets, the better that clinical research variable is going to become. So to really weaponize that, final story, we have built a smart toilet, because why not? So I formed a company called BiomeSense out of the technology, which is currently in China building the prototypes. But the idea is we have a microfluidic sensor system built into the toilet seat, and at the moment a very bulky sample preparation and liquid preparation receptacle just outside of the toilet seat, which allows us to take a stool sample automatically when somebody sits down on the toilet, not invasively. We have a very clever way, it's patented, of catching that material as it passes from the body to the water. And it works whether it's very hard or whether it's very loose. We've worked with all different types of stool. We are not biased to that, which sounds weird. And then we actively collect DNA extract, RNA extract, and metabolite extract that material and store it in aliquots on a chip. And we also have a bioassay, which is still under development and determining whether it's appropriate to apply it, that will allow for on-site identification of metabolites and genetic biomarkers via DNA array hybridization and a very clever system of metabolite identification. And then that information can be sent directly to the cloud. So in this context, we now have a system where we can continually collect stool in a non-invasive way from anybody in a clinical trial, potentially anybody that's interested, that allows us to routinely monitor the microbiota of those people. Why is that important? Well, shit changes, literally and figuratively, right? Every single day we see subtle changes in the abundance and proportion of different organisms and their metabolic potential, and that leads us to be concerned about how a snapshot may actually miss biomarkers of relevance. An interesting work from Ramnik Xavier and Sean Gibbons that just came out demonstrated quite effectively that you need at least 5 to 8 consecutive stool samples to get any kind of good vibe that you've actually detected a biomarker as being statistically present or absent in your individual, right? So just a snapshot has no statistical validity whatsoever. We need continuous longitudinal monitoring. And so hopefully, I don't know if Rob showed these slides as well, but I like these are fun. Hopefully one day, you've done your business on your magic toilet, and bing, when you're cleaning up afterwards, it pops up in the mirror and compares you to everybody else in the publicly available populations, puts you on a microbial map that we can use to identify potential phenomena, like maybe you don't have enough bacteria producing GABA. Maybe your GABA levels have gone down and now you're going to be depressed. I know that happens to Professor Cryan quite a lot. So, you know, what's Professor Cryan going to do when he finally figures out that his GABA's gone to hell? Maybe try some lactobacillus and see if putting that in his body helps. That could be a fantastic way of giving routine advice. And I just want to hold up my hand. I did predict the downfall of uBiome, so I am not in a space to stipulate that this is the right way of doing things immediately, but it's the potential for future activation once we have A, reliable biomarkers that can be detected very rapidly, and C, can actually be used to provide actionable advice, which requires appropriate clinical trials in order to be able to deliver. So I'll stop there. Thank you.